Hey guys, this is Hossein Karnesh, and we're going to talk about the cell cycles of sickle cell anemia and anaplastic large cell lymphoma. In order to understand how eukaryotic organisms have billions of cells, we need to comprehend the process of cell cycle. Cell cycle is a process that all cells undergo to grow and divide, and consists of two phases known as interphase and mitosis. During interphase, eukaryotic cells go through growth and DNA synthesis. Mitosis is a cell division, which is the reason why we have billions of cells, because they constantly divide. Interphase itself has multiple stages. The first stage, G1, is the initiation of cell growth. The S phase is the DNA synthesis, and the final and last stage is G2, which is the final steps of cell growth. In order to understand the inheritance of sickle cell anemia, we must first understand the cycle of meiosis. Meiosis is a type of cell division that reduces the number of chromosomes in the parent cell by half and then produces 4 haploid or gamete cells. The different phases of meiosis are interphase, prophase, metaphase, anaphase, telophase, cytokinesis, interkinesis, prophase 2, metaphase 2, anaphase 2, telophase 2, and finally cytokinesis. Interphase is basically just a cell growth. Prophase 1 is when the homologous chromosomes appear, each made of 2 chromatoids joined at the centromere. Metaphase 1 is the spindle forms and the homologous pairs of chromosomes line up on the metaphase plate. Anaphase is when none of the centromeres are split and the homologous pairs pulled to the opposite ends of the cell. Telophase 1 is when the nuclear membrane reforms around 2 haploid nuclei. Prophase 2 is when the nuclear membrane starts to break down again. The chromatoids are still joined by the centromeres. There has been no DNA replication and there is no crossing over. Metaphase 2 is when the new spindles form and pairs of chromatoids line up on the metaphase plate. Anaphase 2 is when the centromeres finally divide and chromatoids are pulled to the opposite end of the cell. Telophase 2 is when the nuclear envelopes reform and chromosomes uncondense, giving 4 haploid nuclei. Once nuclear division is complete, cytokinesis completes the division process to give 4 haploid cells. In order for sickle cell disease to be passed down from parent to offspring, both parents must pass down the mutated chromosome during prophase of meiosis. This is when recombination occurs, which is where the genetic diversity takes place by switching of genes of the parental chromosomes amongst each other. If both parents have the mutated version of the chromosome, one of the resulting 4 haploid cells that contain both copies of the mutated gene will have sickle cell disease, since it's a recessive trait. Base substitution occurs in chromosome 11 on the codon that codes for the HBB gene, which codes for the hemoglobin protein. One of the alleles in the codon is mutated and alternated, which causes the RNA to code for a different protein. The normal HBB gene that codes for the hemoglobin is GAG over CTC. The mutation of the gene which causes sickle cell disease is GTG over CAC. The A is replaced with a T, and instead of normal production of glutamic acid, it gets replaced with the valine acid, which eventually leads to sickle cell anemia. Sickle cell anemia is a recessive trait. Therefore, both parents have to express sickle cell anemia to have the offspring to be affected. However, both parents are carriers that will also influence the child to be affected. During sickle cell anemia, our blood shape is incapable to carry oxygen throughout our body. Therefore, the symptoms to this disorder are dizziness, fatigueness, low oxygen levels, feeling of malaise, and abnormal breakdown of red blood cells. The treatments of sickle cell anemia include blood transfusions, medications, and rarely bone marrow transplants. African Americans and other individuals that are vulnerable to malaria are prone to sickle cell anemia. Now moving on to anaplastic large cell lymphoma. Cancer arises due to a lack of body's ability to go through natural cell death, also known as apoptosis. Anaplastic large cell lymphoma is when lymphocytes grow and multiply uncontrollably. ALC, also known as anaplastic large cell lymphoma, leads to other complications such as leukemia, heart disease, and lung problems. The treatments are currently being researched by physicians for anaplastic large cell lymphoma. Some medications that help are alisertib, bortezomib, and crizanlizumab. The prognosis of this cancer is that patients need to consistently go in for blood tests and CAT scans to determine if any extensive treatment is necessary. The survival rate for these patients in 5 years is 67%, and in 10 years it will be 55%. The loss of density-dependent inhibition is what leads to cancer production. Normally, if a cell surrounded by many other cells in a highly dense area has a natural ability to stop dividing because there isn't enough space to survive. Cancer cells, on the other hand, divide uncontrollably even if there is a lot of cell density. Similarly, Anaplastic Large Cell Lymphoma loses anchorage dependence, therefore doesn't have cohesion to other cells and grows quicker. In order for a cell to go through mitosis and divide, it must be anchored to something for support, hence the name. Therefore, cancer cells aren't anchorage dependent and divide uncontrollably. The cause for this cancer is when there is a transformation of genes between chromosomes 14 and 18. This causes the mutation of oncogenes. An oncogene has the potential to cause cancer. In a tumor or cancer cell, they are often mutated at high levels. Most normal cells undergo apoptosis. However, activated oncogenes can cause those designated cells to survive apoptosis and instead proliferate during mitosis. This evidently leads to the uncontrollable growth of cells. Anaplastic large cell lymphoma has two subunits. There are systemic ALCL, which affects all organs in the body and aggressively grows, therefore malignant. Primarily cutaneous anaplastic large cell lymphoma is confined to the skin, leading to a slow growth, therefore benign. The growth factor is the protein that code the mutated oncogene, which leads to this horrific, uncontrollable cell growth.